Sorry.
Patients say that voice comes and goes, and I will try to speak to this microphone, and I will try to speak to this microphone, and I hope that it will be heard. And please let me know if there are problems. We follow on Twitter on the Les Turner site. So if there are any problems, please let us know. And it is also taped, and we will make these talks available for each session. So you will be able to watch them after the symposium as well. And now I would like to take some time and to talk to you a little bit about the neuroimmune axis in the motor cortex of ALS. And interestingly, our keynote speaker will also be speaking about the immune system, so this would be a good gateway to her talk. And before I start, I always want to give the acknowledgments in case there's no time to thank the people who are passionately doing the work. And people from my lab, can you please raise up? Barish, I see you. Again, Mukesh, can you please raise up? Mina, and who else? Yes, thank you so much. So we are an ever-growing lab, and today I will be talking mostly about Dr. Gautam's research. But of course, it's not just one team, one-person team. For every project, we have multiple people working all together. And of course, we couldn't have accomplished anything that we have accomplished without our collaborators and with our funding source. So I thank them, and I hope that your name will be on the list soon too. And I will be talking about two stories, but one team. And the stories are about inflammation, about TDP pathology. And these are two recent papers that came out. One of them is in Journal of Neuroinflammation, and we revealed the MCP1, CCR2, and neuroinflammation in the ALS motor cortex with TDP pathology. So we heard about TDP pathology this morning, and we are going to hear more about immune reactions. And I want to bring to your attention that neuroinflammation is one of the common pathways and converging paths for neurodegenerative diseases. Not only for ALS, but for many other diseases, we actually see increased neuroinflammation. But neuroinflammation has many players. And we find that in the cortex of ALS, especially patients with TDP pathology, and involvement of neuroinflammation, and especially MCP1 and CCR2. So I will try to be very brief, and you may not know what is MCP1, what is CCR2, and I can tell you that these are receptor-ligand interaction, and once they find each other, they initiate an immune reaction. So we know that there is increased activated microglia in the motor cortex of ALS patients, and not only ALS patients, this is also observed in Parkinson's disease, in Alzheimer's, in many diseases, that there is increased activated microglia or immune reaction in many neurodegenerative diseases. And in the normal case, I don't know if it works. Yes? Okay, so you can see the bed cells, the large motor neurons in the brain. They have a very long apical dendrite. And you would see, one or two, not very many, astrocytes nearby. But when it is diseased, you will see that astrocytes change their shape, which we call them activated astrocytes, and here, for example, and they also have microglia, IBA1, and we see very long shaped, and then they are usually along the apical dendrites. So we have found previously that the apical dendrites degenerate very early in ALS patients, and that is an important site for neuronal communication, because the upper motor neurons speak to other cells, or they get modulated by their spines in their apical dendrites, and any degeneration at that site means that they will not be properly modulated. And again, the same pathology is observed in ALS FTLD with TDP pathology. So we see increased astrocytes and microglia, and they have a very unique structure. So to be able to understand the underlying causes of this, we actually generated a mouse model in our lab. And previously, the PRP-TDP mouse has been published with by the Balo group. And we had the UCHL1 EGFP mouse model, the first reporter line for upper motor neurons. So we crossed them in an effort to make a reporter line in which the upper motor neurons are GFP and they have TDP pathology. So we will be able to visualize the diseased neurons with pathology in the cortex. And this allows us to see them, see their GFP, within the complex structure of the brain. Even though there are millions and millions of different neurons in the brain, in this reporter line, in layer 5 of the brain, only the upper motor neurons are GFP, and that allows us to visualize them both in vivo and in vitro, and with respect to disease. And if you look, you can see the difference that with disease, there is a lot of activation of the immune system. And we looked at the time, like when does this start? And are there really astrocytes in layer 2-3? And layer 2-3 is important because that is where the most modulation of upper motor neurons take place. So, see, we consider upper motor neurons as a spokesperson of the brain when it comes to initiation and modulation of movement. And many different cell types speak to them, and they get that input, they integrate that input into action potentials to spinal motor neurons. So the place that they get the input is actually around here. That's why the apical dendrite degeneration at this site, or astrogliosis or macrogliosis at this site, can be detrimental. And we actually find that the astrogliosis occurs pretty early, not as people have thought that the brain degeneration is secondary in ALS. It actually occurs pretty early. And there is immense astrogliosis. How about macrogliosis? And you can see, if you compare the two, first of all, there are less motor neurons because they died, because in this disease they progressively degenerate. And not all neurons degenerate, remind you. It is basically these upper motor neurons that undergo progressive degeneration. And there's also increased macrogliosis in layer 5. And that also happens very early. And please look at here, that they actually wrap around them, and it's as if there's a fight between them. See, they're not letting them alone. So there is an immense fight between these microglia and motor neurons, but in reality, they should be friends. They help each other when it is all normal. And we want to understand the molecular basis of this. So I want to emphasize here one thing, that what we see in the mouse here with microglia and astrocytes, and I'm going to show you, this is also in the patients, again, with increased astrocytes and microglia, that they are pretty similar. So if we focus our attentions to the cells and neurons that induce neurodegeneration at a cellular level, the transgenic mouse models, well-defined transgenic mouse models, may actually lead us to understand the cellular biology. Because at the end of the day, we don't want to cure the mouse. At the end of the day, we want to cure patients. And if we can understand what makes these motor neurons upset, then that knowledge can be applied to the motor neurons in patients. And that's our goal. Okay, after we show this phenomena, the question is, who are these cells? Because at first, we didn't have that much astrocytes. We didn't have that much microglia. Did they become activated where they were present? Or did they come from the periphery? So was there a call to pull them to the brain parenchyma? And we have performed multiple immunocytochemistry analysis to see who are these guys that are coming to the brain and who are these cells that are wrapping around the diseased upper motor neurons. And we found that in wild type, you hardly see, if you're lucky, one CCR2 positive cells, and this is a blood vessel, and they are infiltrating from the blood vessel. Every now and then you would see one. But if you look at the disease, you will see 1, 2, 3, 4. There are so many of them. It's almost like whenever you see a blood vessel, there's one cell attached to a blood vessel, and they're coming towards the CSMN. They're very close, and based on the markers that they express, that they are CCR2 positive and TMEM119 negative, that tells us that they are infiltrating monocytes. So they reveal their identity. So they're coming from the bloodstream. So Mukesh actually has done a very good electron microscopy analysis, and he has shown that if he looks in the wild-type mouse, these are the blood vessels, he hardly sees any cell coming from the blood vessel. But if he looks at the motor cortex of the TDP mouse, there are, as you can see, 1, 2, 3, 4, many of them coming from the blood vessel, and they are always in close proximity to these large bed cells. So it's almost towards the end stage. Whenever you see a bed cell, it has a cell attached to it. And who are these, right? The other question is, does this also happen in a human, in patients? And lo and behold, yes, it does. If you look at the normal control, you would see maybe one. Okay, maybe I think you looked more than 50 to be able to see just one, but normally you don't. And look at this in the motor cortex of ALS patients with TDP pathology. There are many, many, many of them, and they're coming from the blood vessel. So we think that they're coming from the periphery. So I think this is an important finding because we did not know, or the field was questioning whether they differentiate right where they are, or there is some infiltration from the periphery. Because if there is, this opens an avenue for therapeutic applications, because maybe we can block them, maybe we can change their minds, maybe there is some way that we can manipulate them. So I also want to emphasize here that if you compare the mouse and the human, the results are almost identical. So I think if we ask the right questions and if you look at the right cells, the transgenic mouse models with cellular clarity will reveal very important information. So, okay, so now they're coming from the periphery, but what is the call? Because you wouldn't leave the blood vessel, go to the brain. Somebody must be calling and somebody must be signaling. There must be a signal. So, because we know CCR2 MCP1 is a good partner for the initiation of innate immunity, first we looked if the CCR2 plays a role. And in the normal control, there's very low levels of CCR2. They express CCR2, but not very high. Here you can see these are the bed cells. But in the diseased, you can see that this is the blood vessel, and look at the cells that are coming in. They are glowing with CCR2. They have very high levels of CCR2. The cells that are coming in, very high levels of CCR2, and they are not TMEM19. And what is the partner for CCR2? MCP1, right? So this is just a drawing to tell you that MCP1 CCR2 interaction is actually the path that leads to innate immunity. And if you look at MCP1, all right, very few levels of MCP1 expression. But if you look at the red cells or upper motor neurons, they have very high levels of MCP1. And MCP1 is a secreted protein, and it is actually a calling protein for the CCR2 positive cells. So now I think we identified a target for the neuroimmune modulation, especially in the motor cortex, that the MCP1 CCR2 are playing an important role for the initiation of immunity. And we have shown you that this starts very early. So in my opinion, this opens a very important research area, especially in the motor cortex of ALS. And I think we should not think that cortex is secondary or cortex degeneration is a byproduct. If we can modulate the health of upper motor neurons, we can have a better impact for the overall ALS pathology. So this is my first part, because then I have shown you that neuroimmune cells infiltrate to the brain. Their migration to the brain starts really early. So this is an early event. It is not a byproduct. And that MCP1 CCR2 signaling is important for the initiation of modulation of neuroimmune reaction in the motor cortex. So this is almost towards I showed you a story that happens towards the end when neuroimmune modulation is at its peak. So now I'm going to tell you something that happens early, or that we think happens early, again, with relation to TDP pathology. And I said, wait, what? Because, again, this is Dr. Gautam's work. We actually made, recently, we made a discovery so unique that we had to come up with a new name, because there was no name in the scientific literature to explain what we have observed. And we called it autophagy, which means mitochondria eating themselves up. And we find that this is a unique self-destructive path mitochondria of upper motor neurons with TDP pathology take, and again, very early in ALS. And I have, on a time scale, I have shown you what happens towards the end. Now I would like to tell you what happens really, really, really early, because we will be talking about pups, meaning P15 mice, which means they are 15-day-old mice, which means their eyes are still closed, they're still with their mothers, they're still lactating, and they're still in the cage. So it's like almost a toddler or a child. It's like, it's not a mature person. This is almost as young as you can investigate in a mouse model. And of course, there's no degeneration, there's no behavioral defects, there's no pathology, there's nothing. They're just little babies. And because we have very many different mouse models for ALS, and which display upper motor neuron pathology, including the SOD1 mice, the TDP mice, which I just showed you, and also the profilin mice. So we have 3 different mouse models, and even though they represent different underlying cause of ALS, patients with profilin mutation and patients with TDP mutation display massive TDP pathology. But surprisingly, patients with SOD1 mutation do not have prominent TDP pathology. And this is an unanswered question in the field. Why would SOD1 patients not have TDP pathology? And why would profilin and TDP mutation patients have TDP pathology? And basically, the TDP pathology is protein aggregations that contain TDP. And you don't have to have a mutation of TDP to have the TDP pathology. You may even have overexpression of wild-type TDP, which would result in TDP pathology. So you don't need a mutation. So it's not really purely genetic. And we don't really understand the mechanisms. But here, to be able to look what is going on inside the cells, inside the neurons, Mukesh has done a very systematic approach. He first labeled them with a molecular marker that is specific for upper motor neurons, CTIP2 labeling. And you can see the ones that are CTIP2 are a bit darker, and the ones that are not CTIP2 are whiter. This is the nucleus, okay? And then he looked at the cells that are CTIP2 positive and looked at their cytoplasm, and the ones that are CTIP2 negative, and again, looked at their cytoplasm. And he realized that there are many mitochondria that are healthy, but there are also many mitochondria that are not healthy. Then he quantified the healthy mitochondria and unhealthy mitochondria, both in CSMN and non-CSMN cells. And I want to introduce the idea of mitophagy to you. Mitophagy is a path that discards diseased or unhealthy mitochondria. It is a very systematic approach because we don't want mitochondria matrix or inside to be exposed to the cytoplasm. The matrix of mitochondria is filled with DNA, RNA, and mitochondrial proteins, and it actually is one of the reasons why innate immunity initiates when the mitochondria becomes leaky. So this autophagy is a very systematic approach where autophagosomes wrap around them and they disintegrate them so that there's no mess in the cytoplasm. Okay, so we actually observe this. There are some mitochondria, here they are with an autophagosome, right? And they don't look very healthy, okay? And they seem to be communicating with each other. And we quantified them both in the CSMN and in the non-CSMN cells. So, this occurs. But in addition to this, we have also observed something very, very unique. And that is the mitochondria. First, there were some mitochondria that were very long and elongated, and they formed a kink like this. Okay, look at the kink. Then they fused, forming ring-like structures. And interestingly, it doesn't end there. We have seen many mitochondria disintegrating from inside out, and eventually the whole mitochondria becomes disintegrated fully. But those are not associated with any autophagosome. It is not mitophagy. So we realized that this is mitochondria eating themselves off, and we called it mitoautophagy. And most interestingly, we have seen them in different stages, mainly in the TDP mice and also in the profilin mice, but not in the SOD1 mice. There was a very striking difference. And we said, okay, what happens in the SOD1 mice if we don't see this? And look, this is what happens in the SOD1 mice. Opa. In the SOD1 mice, the mitochondria is expanded, it's large, and it's most likely the mechanism that these neurons die are quite different. And SOD1, as you know, is superoxide dismutase. And it actually requires more energy to be produced. So then the appearance of mitochondria was very different. You look at the difference. Look at this. It was only really, this was seen mainly in the SOD1. Then, of course, they told us, are you really sure this is not mitophagy? Maybe there is LC3B colocalization. And look at this. If you look at the SOD1 and you do LC3B, which is a marker for mitophagy, okay, for autophagosomes, suggesting that this is mitophagy. And yes, they colocalize. But in the PRP, TDP, you can see those circular ones, you can see those extended ones. They don't really have LC3B. But you may say, oh, immuno didn't work. But if you look, actually immuno worked. Next door, some other cells, they have it. So we believe that this is really a distinct mechanism by which mitochondria degenerate. And you may say, why is this important? And of course, they made the news about this because the self-cannibalizing mitochondria was not seen before. And we think that it may set the stage for TDP pathology to occur. And it may be one of the reasons why we have upper motor neuron vulnerability in ALS. But I want to tell you that this mitoautophagy is different than mitophagy. And the self-eating mitochondria may indeed set the stage for neuronal vulnerability because this happens much earlier than we see any signs of vulnerability in the upper motor neurons. And when Chris was giving a talk about TDP, and we said TDP is in the nucleus and sometimes it's in the cytoplasm, when it is in the cytoplasm, it actually binds to mitochondria. It is colocalized with mitochondria in the cytoplasm. And imagine that this mitochondria is degenerating from inside out. You would imagine that the matrix proteins are all expelled out, and it's not a good thing for the cell. So we would imagine that at that site, it creates hot spots for proteins to aggregate because it's like a glue. And that may be the seeding factor for TDP pathology, meaning protein aggregations with TDP proteins to emerge. And therefore, we think if we can improve the health of mitochondria in ALS, we may also have a potential solution for the aggregate formation. And also, in addition to that, we may begin to have a therapeutic intervention for the neuroimmune modulation, because breaking of mitochondria is one of the early steps for innate immunity to initiate. And we think that mitochondrial problem that we observe here is at the heart of protein aggregation and innate immunity and the immunity that we observe. And if we go to the heart of the problems, I think we will be able to bring effective solutions. So I think we now have a target molecule to modulate neuroimmune reaction, the MCP1 CCR2 axis. And I think we also have a target organelle to improve the health of motor neurons that are going to be diseased. And I think those two are very important as we move forward because these could be both druggable targets and we can begin to think about canonical pathways that would improve the health of the organelle in the system in the cell. So I thank you for your attention, and I'm ready to answer your questions. Yes, go ahead, please. Kristen Di Donato, she has been a very adamant supporter of our symposium, and she has given a talk before, and she's a very good friend. And thank you so much for coming, Chris.
Thank
Kristen.
you. So let's see if I'm still your friend after my questions.
Of course.
Excuse me.
Sure.
So a couple questions. My first one is that you talked about the timing of this early event at P15 in the brain region. Have you correlated that with the timing of what happens in the spinal cord?
No, we have not.
Okay.
You want to do it with us? Maybe we can. Yeah.
Okay. And then the second question is I know that there's been papers out, and you talk about this brain axis of this neuroinflammation, neuromodulation. There's this gut-brain axis that people talk about in ALS and have come out through some of the mouse models. Could you make comment to that? And then I know that there has been manuscripts published on looking at humans to understand if there's a relationship to the gut-brain axis with innate immunity to this. Could you just comment on
Sure.
that?
Before I answer that question, I want to make a comment about the blood-brain barrier. So I think the reason that we have more of these cells infiltrating into the brain regions or CNS in general is because, as you know, the blood-brain barrier is a seal, and it protects our brain. And when that seal is broken, or it's leaky, or it is not as tight, then many cells may pass by. So I think, and many others think, and there are papers to support this, that the blood-brain barrier becomes a bit loose in ALS and also in other diseases. And I don't know the reason why, but this is an observation made by many labs. And I think this adds to the fact that neuroimmune modulation to the CNS becomes a big factor because we don't really have a good seal. And the other thing is, I think, based on our microarray and RNA-seq results, when the motor neurons become diseased, initially they want to maintain homeostasis. And they want to make sure that they do everything in their power to retain their homeostasis. And that's why I think at the very beginning, they may ask or seek help from astrocytes or microglia to help them. Because normally they are there to help. But I think there's a switch, and they say, wait a minute, I can't do this. I can't. It is a switch. Then at that switch, I think that's why we see astrocytes being activated, microglia being activated, because rather than being a supportive, other half, they become detrimental, they become toxic. So there are two parts to it. One is that switch, and the other is accessibility. And that switch, I think, is very molecular, and we are trying to reveal the molecular basis of it. But the accessibility to the CNS, and I think that is mediated by the blood-brain barrier and how accessible the blood-brain barrier is. And that may be a different topic. But I believe that the gut-brain axis is valid. It is true. And I think we should be able to understand the mechanisms to be able to modulate it better, yes.
Your hypothesis is very interesting at the autophagy. But if you want to link this mechanism to the disease, and because theoretically, if the mitochondria eat themselves, and eventually it's a destruction of the mitochondria. And have you ever observed the decrease the number or density of mitochondria at the end stage diseased mouse models, like TDP43 or whatever?
So you're asking the change in mitochondria numbers and
Or
the
density.
distribution of healthy, unhealthy mitochondria
Yes.
with respect to disease progression
Yes.
selectively in those cells. So we are doing those studies, and Mukesh, maybe you want to answer, but I think that this is a dynamic process. And you know, mitochondria is one of the most dynamic organelles in the cell. There's fusion, there's fission, and it is very hard to see mitochondria static, right? And they also move back and forth, even along the axon. You can never see a mitochondria standing at the same spot for a very long time, and they always change their shapes, right? So, of course, when you do EM, it is frozen in time, theta time, and that's what you see, right? And I don't think it is possible to do a live electron microscopy. I don't think it's ever possible. So we wouldn't be able to see how the mitochondria changes over time, especially with the structures that we see with this precision. So that wouldn't be possible. And that's why it's very hard to answer your question. But if we look at the theta time over time with different mouse models, I can tell you that first, the numbers of mitochondria are reduced. Is that correct, Mukesh? And the numbers are reduced, and also we see more of the unhealthy mitochondria in diseased upper motor neurons. Okay, so.
And I have a very basic question. So I think your hypothesis that upper motor neurons are central to disease pathology is very intriguing. Can I ask you, if you selectively rescue using genetic tools SOD1 expression, mutant SOD1 expression in your upper motor neurons, do you rescue disease in the mice? And vice versa, if you only introduce mutant SOD1 or mutant TDP4, you can do this with all your mouse models. Is that sufficient to induce disease?
Yeah, excellent question. So, first of all, I don't want to be seen as someone who says upper motor neurons rule or upper motor neurons die first, or cortex first or something. I just want to say that cortex do not need to be ignored. I think that degeneration happens simultaneously in the spinal cord and the cortex. It's the London Bridge is falling down. Both legs of the bridge is falling down. And I don't think it's just one or the other. And I just want to emphasize the importance of the cortex. I don't want cortex to be seen as a byproduct of the ongoing degeneration. So having said that, we actually have a mouse model in our lab, UCHL1 null mice. And in this mice, the upper motor neurons show very prominent upper motor neuron loss, but not so much the spinal motor neuron loss. And Boris is actually doing those studies, and we are using AAV-mediated gene delivery to incorporate UCHL1 gene into these upper motor neurons to see if we can improve their health, and by improving their health, can we improve also the function of the spinal motor neurons, because they lose their function. And that was a paper that we published with Christine before. And also, if you look at other people papers, for example, when they use AAV-mediated silencing of SOD1 in the SOD1 rats, they not only improve the upper motor neuron health, but they also improve the health of the spinal motor neurons and the neuromuscular junction. Because the order goes from the brain to the spinal cord, and then the spinal cord to the muscle, and spinal motor neurons receive most of their inputs from the upper motor neurons. I think that improving the health of the upper motor neuron may have an added value, and I wouldn't say that's the only thing. It would have added value to improve the health of the spinal motor neuron, because we're talking about the circuitry. And so we're working basically on the UCHL1 model, because SOD1 model has already been published and shown that this is the case. Excuse me? Oh, yes. Okay, yes. Poster number 17 from Barish. Yes, UCHL1 molecule, yeah. All right. So thank you so much. And I was the only person between you and lunch. So please, let's take a lunch break. And unfortunately, our speaker, Eva Feldman, she got stuck in her plane 6 a.m. till 11 a.m. Like 5 hours she was sitting and waiting to be to depart and coming to Chicago. Unfortunately, most of the people in the plane left, but she sent us her slides, and I've been studying them. So me and Dr. Kalp will be giving her talk, and she will also be connected live. So if we make a mistake, she's going to correct us. And if there's more to be added, she's going to add. And I thank Eva for showing this amazing contribution to our symposium. And I also thank you for your attendance for the science session. Thank you so much.